Hello, everybody. Thank you for joining. Thank you for a great introduction. So I'm going to continue the story of the Living Biobank, and I'm a sister core. We deal with in vitro models. These are the next generation in vitro models that are called patient-derived organoid models. I'm sure a lot of you have heard about it. So I will tell you about the collection that we have, about the quality controls, and about how you can actually work with the organoid and also the PDX core. So first, let me sort of introduce the organoid technology very briefly. So organoids are these self-organizing three-dimensional cultures that are generating from stem cells or cancer-initiating cells, or they can also be normal and cancer, and they give rise to specific cell types of the tissue origin. So the first adult stem cell organoids was reported in early 2000s, and it was generated from a cleavers group from a mouse LGR5 positive small intestine stem cells. After this initial innovation, of course, various organoids from different organs and different stem cells have been generated from humans and also other animals. And this is sort of listed here in this review. The two types of stem cells can be used to generate three-dimensional organoids. That can be pluripotent stem cells, but also adult stem cells that are currently, that is the technology that we're currently using in our core. So organoid technology is now utilized in different research fields and has been used as a model to study diseases, gene function, and also cell development. So how do we grow organoids? So really the key to growing organoids is we want to give the right cues to the stem cells or initiating cells in the tissue to recreate the tissue structures. And you do that by basically manipulating the pathways in these cells. So you either activate certain pathways or do you activate other pathways so it's following a certain fate. So these we call tissue niche factors, and some of them are sort of listed here. Really depends on the tissue type. So for instance, with colorectal and pancreatic type of organoids, we do need to activate Wnt signaling, EGF signaling, FGF signaling, that's an example of that. And also it needs the extracellular support matrix, such as the basement membrane, which is sort of the matrigel mixture that supports the generation or the formation of these structures that they're supposed to make in the culture that look like tissues. So you can take, practically the way it works, we take the tissues from the patient. You can take the normal tissue and the tumor tissue, you cut them up in little bits and pieces, and you use some type of collagenase to digest away all the collagens, all the connective tissues, and to leave the tumor cells or the normal cells intact. These cells are then mixed with the matrigel, which is the basement membrane extracellular matrix, and then we put a media on top of these that contain these specific niche factors. And over time, the initiating cells will start forming the structures that will either resemble tumor tissue or the normal tissue, depending on the initial input that you have used to make these models. Now, the organoid technology has wide applications, and that has been used for, first of all, the biobanking purpose that what we are doing. You can use them for targeted gene editing. You can use them for disease modeling. Because they're pure tumor cultures, they're also very good for profiling. So, you don't have the connective tissues, you don't have other cell types that you can find embedded within the tumor tissue if you are doing this directly from the patient. They are, I always say, whatever you do with cell lines, you can do with properly QC'd organoids. They grow in large numbers. We could do a lot of drug screens, for example, that are 1536 well-played drug screen, so they can be adapted to any kind of high throughput screen. And you can also study host microbiome interaction. So there's been a lot of work done on, for example, using colorectal organoids to understand the interactions with the microbiome within the gut. Now, there's several groups that have published generation of organoids from different tumor types. And this is sort of shown here in this table that's been published in one of the reviews. And what you can see that, really depending on the cancer type and the source of the tumor. So, even you can start with a surgery piece, you can start with a biopsy or a fluid. The efficiency rate of establishing these models varies. Now, the problem has been, when you read these articles, what is really defined as efficiency, and that as a living biobank for us, that is very important because we want to make a model that we can give it to our researchers and they can continue working with them, and they're basically renewable models, just kind of like a cell line. So the efficiency rates reported in different ways, really depending on what article you're reading. But it could be, for example, some people say organoid initiation. So whatever really grows, whether or not any organoid growth after tissue processing would be an organoid initiation. And actually, a lot of the papers report this as a success rate. Short-term cultures are the cultures that you can propagate a few times. They'll grow in passage 1, you split them, they grow in passage 2, and they start slowing down. Passage 3, passage 4, they don't grow anymore. As a biobank, perhaps depending on your input, like if you're starting with a lot of tissues and you can work with some short-term cultures, if you do a proper quality control, perhaps you can do some experiments as a researcher. As a biobank, we cannot work with those. We cannot we cannot freeze them down. We cannot revive them. We cannot expand them in large enough numbers to actually share these models. So the only efficiency that we are concerned is the long-term culture. So these are the cultures that will continue growing beyond passage 10. And again, is similar to having a cell line, with a proper protocol that will continue growing for you, and you can sort of predict the growth and the behavior. So now that we know what we defined as a model in our hands, so the following is the workflow that we actually follow from receiving the tissues to generating these organoids to biobanking them. So you can see that we collect a large amount of data at each of these steps, and that's all properly documented. So we can, go back and calculate our success rates, and we can sort of troubleshoot and develop our protocols a little bit better. So we, in pre-processing stage, once we pick up the tumors, we will always save a little piece for FFPE, for histology sample. We also snap freeze the tissue if we want to later on do SDR profiling, or if we want to do any other omics profiling. Then we process the tissue, we count the number of cells, and we estimate the live number of cells and the sample viability. And then we would plate these cells in, we usually use 24 well plate format. So we mix them with Matrigel, we plate them, and then we use different medias. Now the medias is different depending on the tissue type. And very often we use multiple medias for the same tissue type. So for example, for growing colorectal organoids, we don't have a single media. We initially start with 6 different medias, and then we pick the media that the model grows in the best. So, all our passaging ratios are recorded, how often we passage. We also take images along the way. And once the model actually makes it, and that depends on the tumor type. So for instance, we're more aggressive types such as pancreatic organoids, we know that if they make it to passage 5, they will make a long-term organoid model. So at that point, but for example, for breast and lung, one of the problems with those tumor types is that there's very often a normal outgrowth. So even though you think you're growing the tumor organoids because you started with the tumor tissue, they could turn out to be normal outgrowth. So we really have to passage them much longer and also do more QC to make sure that we're growing tumoroids and not normal organoids. So once we know that the model made it, then we do a lot of quality control assessment. So for example, we do histology analysis where we compare the morphology of these organoids to the original tissue. We do mycoplasma testing on all our frozen stocks. We perform doubling time, which is very useful measurement if you're doing drug screens, for example. We also have passaging data, so we know exactly, this model is passaged every 10 days, 1:4, 1:6 ratio. Again, this is depending on who's working with these models, but that's like a general guide on how well they grow. Also, if we're starting with xenografts, and we do make a lot of organoids from nuens, PDX library, one of the issues is in some cases we can have mouse outgrowth. So, instead of growing human tumors, the mouse tissues will start overgrow. So that's why we do flow cytometry to make sure that whatever we're growing is the human tumor cells. We also do STR profiling to make sure that the model matches the patient. SNP profiling is used to assure that we don't have normal outgrowth, that what we're growing is the tumor organoids. We also inject these in mice again, that's another test to confirm that these are tumor organoids. And then all our samples are submitted for omics data. So we do whole exome sequencing, shallow whole genome sequencing, RNA-seq, and for some samples, we also have ATAC-seq profiles. They're biobanked, so we do cryopreserve them at different passages, and we also perform the cryo regrowth to make sure they revive properly. So as you can see, there's a lot of work going on to trying to get these models to be I guess successful in everybody else's hands. So we're very supportive in helping whoever gets these models to start their work. So this is examples of some of the tissues that we get. Again, we either start from surgical resection, xenografts, biopsies, or we can also work with fluids, such as pleural fluids from lung, and also ascites fluids from ovarian patients. So once these tissues are processed, we'll plate them in the Matrigel, and you can see there's an example of one of the wells. This outline here shows the Matrigel, and you can see that the cells are sort of individually plated at the time of dissociation, and after 7 days or so, they sort of start bubbling up and they start forming organoids. So when we looked at these organoids, the properties, they're really patient-specific cultures. They resemble in vivo complexity of tissue architecture. So the cell hierarchy, you can see some of the organoids here. These are just your bright field images, and these are H&E histology images. So you can kind of see that there's definitely polarity in these organoids. They're forming lumens just like they would in a patient's body. Some of them have multiple lumens, and there's a lot of different morphology even within the same organoid culture that comes from the same patient tissues, which kind of it's similar to the original patient. So they do also recapitulate and preserve the genomic complexity of patient tumors. They don't really change a lot in terms of as you passaging them genetically like the cells do. And they can also be cultured again from a very small sample size. So you can study heterogeneity of tumors. If you, for example, make organoids from different parts of the tumors, that is definitely feasible. So organoids mimic tissue cell hierarchy. So I'm going to show you a few examples here. This is an example of a lung organoid that's generated in our core here. This is a normal lung organoid. And we know that the lung organoids or lung airway contains differentiated ciliated cells. So the cilia formation is labeled here as red because we stain it with the beta tubulin 4. This is a control, so you can see the blue cells are just the DAPI stained nuclei, and the red are cilia. And in organoids as well, you can actually see cilia formation on 2 different models. That sort of speaks to the differentiation of these organoids in the cell culture. Here's another cell type. So this is mucin. So the lung epithelium does contain mucinous cells, and this is 2 controls. This is the normal lung epithelium, and this is the mucinous tumor. And then you can see our organoids also contain some of the cells that are mucinous. These are some of the images from the organoids that we have generated in our core, some of the H&E slides, and also immunohistochemistry. So we do get pathologists to actually look at each one of these models and to confirm that they match the original tissue, and we stain them with the antibodies that are used in the clinic. So for example, for a lung adenocarcinoma models, we use a TTF1 stain. So here is an example of an organoid model that was made from this particular tissue that is TTF1 positive, and so is the organoid model. Some other examples are, for instance, esophageal adenocarcinoma. You can see the positivity for p53, both in the tissue and in the organoid. So this is our inventory. This is basically our collection. All of the models are long-term organoid models, and all of them have the quality control approved, which means all the tests that I mentioned before, these models have passed. So we currently have 263 right now, probably even close to 300. I would say that we bank 50 long-term organoid models a year. That is sort of our, might not sound like a lot, but this is a lot of work to actually get these models properly done. And also because of the success rates are not always the same for all the tissues. So you can see we also get PI support, both through helping us develop the SOPs, but also providing some of the grant funding. And we worked with a lot of clinical trials groups as well, or the clinicians. So a lot of the models that we make are quite clinically relevant models. So they're not just models that are made from surgical tissues and naive patients that have been treated. We have organoid models that are made, for example, from patients who are resistant to certain treatments. So these are very valuable models. So example would be, for example, lung patients who are resistant to osimertinib. So we have generated models for those particular patients, or ovarian organoid models from the patients who are, for example, became resistant to PARP, to, for instance, olaparib or any kind of PARP therapy. So our biggest collection is pancreas, because that is the first set that we started with. We have a large collection of colon and the lung, followed by esophagus, ovary, and breast. You might ask, why do I have such a small collection of ovary and breast? If you have worked with this tissue type, you will know that these are very difficult to make. The success rates are less than 10% for long-term organoid models. And while we have processed hundreds and hundreds of different specimens and tested many different medias, they're still biologically very difficult to make. So, even though you have a small number, these are quite valuable organoid types. Each model, if you're working with us, we do, and I'll tell you a little bit about the services that we have, but each model comes with its own little characterization sheet, and where the model comes from, what was the patient's original basic demographics. We also tell you what the histology looks like, what is the passaging ratio, what is the days between passaging, and what type of media. If you're working with us and if you get our models, we also do provide detailed SOPs, which include propagation, media recipes, freezing, and we also have training for this if anybody is interested. What can you use them for? So there's many applications, as I mentioned before, for using organoids, but one of the first applications that we have done is organoid drug screening. So you can use organoids. As I said, you can do drug screening in 384 well plates, but you can also do high throughput drug screening in 1536 well plates. So we have done both. But basically, it depends on the drug. It depends on your experimental protocol. But here I'm just showing you that what we really do. For instance, we would dissociate the organoids to single cells. We'll plate them in the 384 or 1536 well format, and then we would add the drug, whatever it is, chemo drug or targeted therapy or epigenetic drug. And then after a certain amount of time, we could measure viability. So we could do an endpoint assay, for example, ATP-based assay, or we can also do imaging or apoptosis-based assay, whatever really depends on your on the drug and on your research question. So the first set of drug screening data that we have done was for the original COMPASS study. So the COMPASS study was a large clinical trial led by doctors Jennifer Knox and Stephen Gallinger here at UHN. So they collected clinical and molecular data from patients who had advanced stage pancreatic ductal adenocarcinoma, and the generating PDOs was just one arm of the study. So the goal of the study was to generate pharmacotype PDOs from patient biopsies and correlate the therapy responses in 20 patient PDO pairs. So we would make the organoid model, we would drug screen it, and we would get area under the curve, calculate area under the curve for each of the organoids. So basically, it's showing you here anything below the median would be the organoids that are sensitive to that particular drug. Anything above would be the resistant. I mean, you can even go further and you can look at, quartiles. Really, again, depends on your initial experimental setup. And then, once you group your PDOs, you can then correlate the response. So if the patient is, for example, sensitive to gemcitabine, organoids should really fall within this category. So that sort of, because we still don't know how well these things correlate. So this has been currently this is being published, but this is an abstract at that was done at AACR. And you can see some of the IC50 curves that are generated for each one of these drugs. And then this is the area under the curve. So overall, when we do look at the correlation, and others have reported this, some drugs correlate more than the others. So for example, there's more correlation with a drug such as paclitaxel, but there are other drugs that might not correlate as well, and some examples would be platins. So platins overall haven't really been a super correlative with when you compare the patient and in vitro model, and that's probably because of the way that these drugs are even handled. The platins really don't have a very long shelf life, so that could be one of the problems there. But irinotecan is another one that seems to be correlating very well. So that's about the organoid core. Both a subset of PMLB models, both PDX and PDO models, are currently visible on cancermodels.org database, which is supported by the National Institutes of Health, National Institutes of Health and National Cancer Institutes, the Jackson Laboratory. So this database contains the list of models from different institutions across North America and Europe, and it's very useful. Also, our models are featured on Biosample Hub, and they can be accessed through there. We have worked, our user community is definitely expanding. We are using, we work with a lot of academic institutions. We work with nonprofit research centers such as OICR, but we also work with commercial entities as well. We have published our organoid sets and the PDX sets, both with external investigator and as well as the UHN investigators. And you can definitely look up some of these publications. So how can you work with us? So first, model generation. So if you're interested, for example, in generating models from tumor types that are currently not in the biobank, you can certainly contact us and we can talk about that. We're always looking for tumor types to add to the biobank. We also can help with the grant support. We do distribute these models under MTAs internally and externally. If you're interested in getting some of these models from us, we can certainly start the process with you. We do if you get our models, we will help you with the SOPs. We also have organoid consumables. So we do make things like organoid media or Wnt and responding medias. We have done that kind of stuff. Also, with drug screening projects, a lot of the models that we make, we work with the PIs and perform specific drug screens on them. And you might have an existing model. Perhaps you need help in quality assessment of these models and any kind of custom projects that you can sort of think about. So this is my last slide, probably the most important slide. Again, we have been around for 8 years now. It's a large effort. It's not just, the core technicians are Nguyen and myself. It's really the support of the whole UHN community. Our director, Dr. Ming-Sang Chow. We also have scientific advisory committee that comprises of different PIs that are sort of interested in this work. We have really great set of technicians that work with us. We also support a lot of projects of like the different postdocs and graduate students. And we also work with supporting UHN course that includes the Animal Resource Center, our biobank, the biomarker labs, cancer registries, and so on. So I'm gonna finish with this, this is sort of our email. If you're interested, you can email us. We can talk more about maybe getting some of these models or helping you with your work. And I'll welcome any questions.
Okay, thank you very much, Nikki and Nguyen, both of you for 2 wonderful talks. And so look forward to seeing if there are any questions now. Okay, here's one question. Can you see that, Nikki?
I can see this, yes. So Jonathan is asking if I can generate xenografts from organoids. Yes. So one of the tests that we do, so for example, if we are just making a PDO, which is patient-derived organoid directly from the patient tissue, one of the tests to make sure that this is actually a tumor organoid is to inject it in mice. So we have found that they grow very well. If they're tumor organoids, that will grow very well in a xenograft, right, Nguyen? We call those ODX, organoid-derived xenografts. So the names are confusing. We have PDOs, patient-derived organoids, XDOs, xenograft-derived organoids, and ODX, organoid-derived xenografts, right? So the names are getting very confusing.
Right.
Oh,
another question?
Yeah, so Kenny's asking, when you take a specimen all the way through to the end, roughly how many banked vials do you end up with? So that's a good question. I mean, you can bank as many as you want, depending on how long you're growing them. But we typically cryopreserve 12 vials at different passages. And those are sort of our banked vials. But let's say somebody is interested in getting the models or we need to expand it, then we would take one of those vials and then expand it further. But you can, as I said, these are long-term organoid models. You can freeze them down every week if you want to.
Okay. Thanks for that question, Parag. Do you want to speak or?
Yeah, we follow the same strategy for the xenograph level in terms of banking. We like to test our models up to at least 3 or 4 passages. The more difficult they are to grow, for example, if you're reviewing the histology at the endpoint and they have low cellularity, you like to bank more vials rather than less and over different passages. For sure, within the first 1 to 5 passages, the more you bank, the less likely you'll be depleting your specimens for future use.
Right. Okay. Thanks, Noel. Can I just say, before you leave the platform to everyone, please take a quick look at the expo section. I think you might find that interesting. All right, thank you. We have a question from Peter Lynn.
Yeah, so the question is how amenable these organoids to genetic manipulation, such as inducible expression. If you take anything from this seminar, I would say whatever you can do with cell lines, you can do with properly QC'd organoids, like it's going to work. So we have done Cas9 knockdowns, so we have done siRNAs, like it actually works fine. You just need the proper protocol, for instance, for transductions. So the transduction is a little bit trickier, but it's just a matter of using the right protocol, so it will work.